Up next, we've got a presentation on a mesocosm and field study of tape grass, capacity to sequester nutrients and suppress algal growth. Dr. James Douglas will be presenting this. He's from the Water School at the Florida Gulf Coast University. The purpose of this is to provide information about a CHNEP funded research project that analyzes the capacity of tape grass meadows to improve water quality. This relates to water quality improvement, fish wildlife, and habitat protection priority areas of the CCMP. So with that, I will hand it over.
Okay, thank you all for the opportunity to do this research and talk about it. These results that I'm talking about today are very fresh. Sometimes you hear the term hot off the presses, but this data is still wet. And this is somewhat of a disclaimer because being so fresh, we've got some more questions than complete answers here, but what I've done my best to bring all this data together and try to answer our initial questions as best I can at this point. While we're on this introductory slide, I should acknowledge the other PIs of this project, my colleague here at FGCU, Wynn Everham, and scientist at Johnson Engineering Incorporated, David Seeley, also helped design and carry out this project. And I need to give a big thank you to my graduate student, Brondum Krebs, who as soon as he enrolled here at FGCU, he was immediately tasked with a lot of work to help get this project done. And he did a fantastic job with this really, really large task of setting up a Mesocosm array and carrying out dozens of different nutrient analyses. And then just this summer, doing it all in the field, which is what you see in this picture here, where these very shallow water divers, Brondum is the one with the blue tank in the center, are analyzing the results of our field experiment here. This project involved both a mesocosm experiment in tanks and an experiment in the Caloosahatchee Estuary. In addition to this graduate student who was really the linchpin of the project, we had lots and lots of help from FGCU undergraduate students and volunteers. You can see one of our champion undergraduates, Lauren Tracy, attached to the floating cooler there in the picture. All right, next slide, please. So what I'm going to talk about today is a little bit of background on the Caloosahatchee Estuary and the story of its SAV loss and restoration needs. And I'm going to talk about some of the motivations for restoring seagrass using what we call the ecosystem services concept, the idea that features of the natural environment actually provide valuable services to humankind, including nutrient sequestration. We'll talk about the methods that we use to try to quantify the ecosystem services of the restored SAV in the Caloosahatchee. And those methods involved a mesocosm experiment, one in tanks, and the field experiment. That's the picture we were just looking at in the estuary. I'll give the results, focusing mainly on the results of the mesocosm experiment because we're still getting some of the results from the field experiment back from the lab. I'll talk about the impacts of nutrient enrichment that we saw and some of the effects of SAV presence or absence. And I'll do some back of the envelope calculations to try to extrapolate what we saw in the experiment and field studies to a greater understanding of the role or benefits of these underwater plants in the Caloosahatchee Estuary. And at that point, I'll sort of open it up to a discussion or critique as the case may be of the results. So next slide, please. This is not new news to any of you, but I thought I'd start with a basic introduction to what submerged aquatic vegetation is. SAV is a term that can apply both to underwater plants in saltwater, which we call seagrasses, and underwater plants in freshwater, which may look like seagrasses, but just fit into a more general category that we call SAV, for lack of a better word. So an example of a seagrass, which is a saltwater SAV in the Charlotte Harbor region, is turtlegrass, Thalassia testudinum, and freshwater lookalike to turtlegrass is tapegrass, the scientific name Vallisneria americana, which was the focus of this study. So this is a freshwater plant, but it does occur in our estuaries in the oligohaline regions where the water is mostly fresh most of the time. This is a really important part of our estuaries for multiple reasons. One of the reasons, can I get a click? One of the reasons is because manatees move between the higher salinity and lower salinity habitats in our estuaries on a seasonal basis, and manatees eat both the seagrasses and the freshwater SAV. Unfortunately, although as we learned about in Mark's presentation, we're losing seagrasses from the high salinity regions. We're also losing these freshwater SAV from the low salinity regions of our estuaries, and that's a particularly big threat to the manatees in the cold season when they swim upstream to look for warm water refuges and alternate food sources in the cold season. So both of these types of SAV are really, really important for feeding manatees as well as providing other services to nature and humans, as I'll talk about. Next slide, please. So the Caloosahatchee Estuary, like all estuaries, spans a gradient from fresher waters to the salty ocean. And along that gradient, there's a change in the submerged aquatic vegetation communities from these freshwater plant communities in the upper part of the Caloosahatchee Estuary. This is a figure from South Florida Water Management District monitoring, which shows the approximate different SAV zones of the estuary. And what you see is sort of in the Fort Myers area and upriver of that, you have this Vallisneria americana freshwater SAV zone. In the lower part of the estuary, including San Carlos Bay, Pine Island Sound, Matlacha Pass, you have these mixed seagrass beds of turtlegrass, Thalassia testudinum, and shoalgrass, Halidule wrightii. And then there's, in the lower part of the estuary, it's a little too fresh for the turtlegrass, but you do still have shoalgrass. This diagram, if you look at the red shading, this is sort of the historical habitat of the tapegrass. And there's actually not tapegrass in all of this area anymore. In fact, now the tapegrass is found just sort of from the points 1 and 2 and up in the estuary. Due to salinity intrusions, the tapegrass has been wiped out from the area further downstream in the estuary, and it's quite sparse in the upper estuary as well. Next slide. This will just put a little pictorial representation of what the dominant seagrasses are in these different regions of the estuary, from the turtlegrass towards the ocean, the shoalgrass in the middle part of the estuary, and this tapegrass, Vallisneria americana, in the upper estuary. Next slide. This is some snippets from a recent publication that I helped produce with co-authors from the South Florida Water Management District. We put together a lot of the district's water flow data along with their seagrass monitoring data to construct a time series linking the trends in hydrology in the Caloosahatchee Estuary and SAV abundance in the Caloosahatchee Estuary. And in this figure from the paper, you can see the hydrologic trends in the gray line and the trends in tapegrass Vallisneria in the black line. So where the gray line goes up is where salt water crept up the estuary into the tapegrass habitat. And you can see that after these spiky intrusions of salt water, we lost the Vallisneria. And there were sort of repeated cycles of this beginning around the year 2000 and continuing to the present. And they just sequentially knocked down the tapegrass from near 100% cover at some of the reference sites where you had 400 plus shoots per meter squared of tapegrass to very little tapegrass at all in this region of the estuary. We could say that the tape grass is ecologically extinct in the estuary now. There is some, but it's so sparse now, it's really not doing what it used to do for the estuary. Next slide, please. We have recently, through another project run by a combination of FGCU researchers and collaborators from the Sanibel Captiva Conservation Foundation, updated our estuary-wide map of seagrasses in the Caloosahatchee from I-75 to the Sanibel Causeway. And this is to sort of give us a picture of the underwater environment in the Caloosahatchee Estuary that we unfortunately don't have from aerial photography. Because the Caloosahatchee Estuary is so tannic and murky, it's really not possible to do aerial photography monitoring for much of the Caloosahatchee Estuary. And so we had this many years of gap in our understanding of really where the seagrass was in the Caloosahatchee Estuary and how much there was and what kinds there were. And so with this project, we did 100 shoreline perpendicular transects, very similar to the transects that Melinda Brown was talking about from the FDEP's monitoring, except that we just did tons of them. And the idea was that by having transects all up and down both shorelines of the Caloosahatchee Estuary every kilometer, we could sort of generate a map of where all the different SAV species occur. So I'm only showing two of the SAV species, the two that I mentioned earlier, the saltwater flagship SAV, which is the turtlegrass, and you can see that that's concentrated in the lower estuary, the San Carlos Bay area, as indicated by these green dots, which are sized according to the percent cover of SAV that was observed in the area there. And then you have to look a little bit more carefully in the right-hand panel to see where the tape grass Vallisneria can be found. It's found only above Fort Myers, and it's not as abundant, not nearly as abundant as the turtlegrass is further down in the estuary. So there are a few sites in the upper estuary where we can find some tape grass, but it's just not as abundant as it used to be, or nearly as abundant as the turtlegrass is in the lower estuary. Next slide, please. So because the tape grass has really declined from where it used to be, there's been a lot of focus on bringing it back. This is a picture of my collaborator, David Seeley from Johnson Engineering. Behind David, you can see these round cages, which are a useful method for restoring the grass. One of the difficulties that we've encountered as we try to replant the grass is it's really hard to bring it back from nothing. It was much easier to lose the grass than it's been to bring it back, because when there's a small replanting of seagrass or tape grass, it tends to attract all of the grazers in the area, and it's hard to sustain. But the idea is that by replanting the grass in these cages that protect it from grazing, we can increase our likelihood of getting a successful starter population of the grass, which we then hope will spread beyond the cages and repopulate these parts of the estuary that have lost the tape grass. David has been doing these restorations both in the tidal brackish part of the estuary and above the S79 lock and dam in the freshwater C43 canal. One of the benefits of this restoration is an improvement in fish nursery habitat, and that's what this visual kind of emphasizes. These are fish that Dr. Seeley has trapped in the vicinity of the cages, and FGCU researchers have investigated the fish communities in and away from some of these restoration areas and found some intriguing differences in fish communities where the grass is being restored or not, which suggests that provide evidence for one of the hypothesized ecosystem services of tape grass restoration, which is an improvement in fish productivity and diversity. But improving fisheries habitat is just one part of the justification for this effort of restoration. And I want to talk about some of the other hypothesized benefits of restoration on the next slide. So improving fish habitat is one type of ecosystem service that can be provided by SAV. But let's zoom out here a little bit and talk about this general idea of ecosystem services. Like a lot of ecology, there's jargon that we like to throw around, and ecosystem services has a specific meaning that we need to stop and define for a second. An ecosystem service is a benefit to humanity that is provided by the functioning of an ecosystem. So an example of an ecosystem service would be something that provides goods to humans, like a functioning ecosystem could provide plants or animals for consumption. It could provide wood for paper or building materials. It could provide oxygen to breathe. So that's an ecosystem service in the form of providing some substance. And then another type of ecosystem service is the idea that ecosystems regulate the environment in ways that help us. They clean the water, they protect the soil from erosion, they can moderate the climate by sequestering carbon or providing shade. So in order for the ecosystems to provide these valuable services to humanity, we need to make sure that they're cared for, that they the key species are protected and restored, and that the biodiversity is maintained, because many studies have found that there's a link between biodiversity and the presence of key species and the ability of ecosystems to provide for us. If we don't take care of the environment, it doesn't take care of us. Next slide, please. This slide lists some of the ecosystem services that have been associated with submerged aquatic vegetation through other research. One of the ecosystem services is the primary production of plants and algae that occurs within these seagrass beds. Seagrass beds are incredibly productive, and that productivity of plants and algae transfers up the food chain to productivity of invertebrates and fishes, including valued game fishes like the snook and tarpon that were mentioned earlier today. Another ecosystem service of SAV that's gotten a lot of attention in recent years is carbon capture and storage. Seagrass takes carbon out of the water as it grows, and that carbon, much of it is stored as organic matter buried in the sediments beneath the seagrass bed. And this removes carbon from the ocean atmosphere system and can help counteract the carbon dioxide enrichment of the atmosphere by human activities, lessening the impacts of climate change and ocean acidification. Submerged aquatic vegetation can also stabilize the sediments, preventing erosion and preventing an increased turbidity of the water. Seagrasses can absorb nutrients from the water and take them out of the water. And through competitive nutrient uptake, basically, if the seagrasses take the nutrients, then there's less nutrients in the water for algae, including harmful algae blooms. In addition to stealing the nutrients that would otherwise fuel algae blooms, seagrasses can sometimes suppress algae through chemistry. The seagrasses will exude chemicals that suppress algae growth. And the combination of these last 3 points, sediment stabilization, nutrient sequestration, and allelopathic suppression of algae, are some of the ways that seagrasses lead to improved water quality. Next slide, please. So we had these sort of hypotheses going in that seagrasses were going to have these benefits. But we in Southwest Florida here, we really didn't have much hard data on the capacity of seagrasses to do these things. And so those were the objectives of our study. One, to determine how much nutrients the Vallisneria americana in Southwest Florida could be removing from the water. To sort of extrapolate that to the restoration effort and see, by restoring SAV, how much more nutrient removal could we get? And then once we had that information, share that with the public, because translating environmental needs to human action is always a difficult thing. And if you can cite some statistic that the public can hang on to, that can be really helpful. People always latch onto this idea of trees provide oxygen that we need to breathe, and or one oyster can filter 50 gallons of water per day. Those kind of statistics are really helpful, and people remember them, and they can be used to sort of improve public support for conservation efforts. So we wanted to get some statistics like that we could use for to bolster the restoration effort in Southwest Florida. Next slide, please. The two approaches that we used for this were a mesocosm study and a field study. Mesocosm is just a fancy word that scientists use for bucket or tank. And we used two types of mesocosms in this study. The kind that were in our original design were these large mesocosms. They're cattle tanks meant for cows to drink out of. 416 liters, big tanks, which we set up 20 of these big tanks outside at one of Florida Gulf Coast University's research facilities, the Emergent Technologies Institute, which is on Alico Road near Quarry. And then we got interesting results from that experiment, but we had some regrets about how we'd done certain things, and so we're currently doing a follow-up to that experiment with more but smaller mesocosms, which we've established in buckets. So that's currently underway. So I don't have results to talk about from the bucket experiment, but I'll be talking about results from the cattle tank experiment. And I'll also be talking about some of the results from the field experiment that we did, where we manipulated our grass presence and absence and nutrient addition in the Caloosahatchee Estuary rather than in buckets. And we did that just this summer, which is why some of the results aren't back yet. Next slide, please. This is what the Mesocosm Array looked like at FGCU's Emergent Technologies Institute on Alico Road. You can see it's an elaborate setup where these cattle tanks are balanced on cinder blocks, on pallets. There's a air pump manifold in the center, which is bubbling air to all of these things. They're also covered by shade cloth, which blocks 50% of the light. Otherwise, they would be unrealistically bright. And not correctly simulating the underwater light environment of the Caloosahatchee Estuary. In these tanks, in these 20 tanks, we manipulated two factors: the presence or absence of Vallisneria. Some of these tanks were planted with 30 shoots of Vallisneria, others received no Vallisneria, and the presence or absence of nutrients. At the midpoint of the experiment, we added nutrients to some of the tanks and not to the other half of the tanks. And so the combination of these two factors, each with two levels, resulted in 4 unique treatments: an ambient treatment with no nutrients or grass, an ambient treatment with no grass but nutrients, a treatment with grass but no nutrients, and a treatment with grass and nutrients added. Each of these treatments was replicated 5 times, which is why we had 20 tanks. Next slide, please. The type of SAV that we used in this was Vallisneria, but not all Vallisneria is created equal. We wanted to be as realistic as we could be, and we therefore used the Caloosahatchee strain of Vallisneria. If anyone has been out in the Caloosahatchee Estuary recently, and has seen the Vallisneria in the Caloosahatchee Estuary, it unfortunately doesn't look like the Vallisneria in the picture here. It's very short. The shoots are usually less than 10 centimeters tall. But that's the species that we have in the Caloosahatchee Estuary. That's the one that seems to be able to tolerate the low light and sometimes high salinity conditions in the Caloosahatchee Estuary. And so that's the one that we used in this mesocosm experiment. We enriched the waters in these mesocosms with slow-release granular fertilizer. This is a type of fertilizer that's commonly used in seagrass studies, usually in the field. And I've got this 100 grams highlighted here because my greatest regret about all these aspects of the experiment that we did was the amount of nutrients that I used. We used too much nutrients, and we caused hyper-eutrophication of the nutrient treatments. This would have been a good amount of nutrients to use in a field study, and it is the amount that we used in the field component of this. But we succeeded in increasing nutrient levels in the mesocosms, but we were a little too successful there, and we'll talk about that later. Next slide, please. My graduate student, Brandon, was fantastic about monitoring the environmental conditions and water quality in these mesocosm tanks over time. So we deployed light and temperature sensors in the tanks to make sure we were accurately simulating the habitat of Vallisneria in the tanks. And we also monitored many water quality variables at several points during the experiment. And we monitored the response of the grass in terms of its abundance, in terms of numbers of shoots, height of the shoots, and at the end, the total biomass of the shoots. And also, we wanted to know where the nutrients that we were adding to the water ended up. Were they getting sucked up into the blades and roots of the grass? Were they ending up in the phytoplankton or the sediments? So we analyzed the solid materials nutrient content of those things. Next slide, please. The field experiment had a similar factorial design where grass presence was manipulated, or, well, we didn't take grass out or add grass in. We just made use of the fact that we found an area in the upper Caloosahatchee Estuary where a fair density patch of Vallisneria was adjacent to an area of the same depth and sediment type without Vallisneria. And so we were able to establish an array of plots within the bed and outside the bed, and we were able to do nutrient enrichment at some of the plots in each of those areas to create our 4 treatments. We monitored nutrients in a similar way, and we did the same kind of nutrient dosing with 100 grams of slow release fertilizer in a little sort of tea bag attached to a stake in the sand, and monitored the water column and other materials, nutrients over time. Next slide, please. This is a little picture of our follow-up experiment. This is what you might call Mesocosm experiment Mark II. And this is an overhead shot where we're looking down into 6 buckets. We use 6 different levels of nutrients in this experiment, and none of those levels of nutrients was as high as what we had used in the original tank experiment. We wanted to see if we could capture sort of the point where nutrients, instead of helping the grass and the ecosystem, switched over to causing this harmful eutrophication. And we'll soon have results from this experiment as well, but we don't have them here. One reason that I did want to show this slide, though, even though we don't have the final results from this experiment, is it shows the sort of small shoot clusters that are typical of this Caloosahatchee strain of Vallisneria. It also shows these light green ribbons in the tanks. These we put in the tanks as a way of measuring the growth of epiphytes, and we did that in the other experiments as well. Next slide, please. So I'm going to talk about the results of the experiment, looking first at some side-by-side comparisons between the Mesocosm experiment and the field study, and then focusing mainly on the Mesocosm study because that's where we have most of the complete data. One of the differences between the Mesocosm study and the field study was that we did the Mesocosm study in the fall. Water temperatures averaged around 20 degrees Celsius in the Mesocosm experiment, whereas in the field study we did that this summer and water temperatures averages around 30 degrees Celsius. There was also less variation in water temperature in the field because of the large mass of water, versus in the Mesocosm study you had some day-to-day ups and downs and some week-to-week ups and downs as different weather systems were coming through in November and December of that experimental period. Next slide, please. These are data from the light loggers that we had in the experiments, and they show photosynthetically active radiation on a 24-hour average photosynthetically active radiation within the tanks. You can see that it goes up and down from day to day as you have sort of cloudy and sunny days. But in the Mesocosm experiment, the average is maybe 60 or so micromoles per meter squared per second over the experiment. That is enough. That's plenty to sustain Vallisneria. There was enough light in the field as well, but the overall amount of light in the field was less, and also because this field experiment was put in kind of at the beginning of the wet season when flows from the Caloosahatchee were just beginning to increase. We saw a decline in light over the course of this experiment as we moved into these wet season conditions with a big influx of highly tannic and turbid water. Some of the light sensors in the field experiment were occluded by drifting macroalgae, which was something that we encountered in the field. And this is something that the grass would have to deal with as well. Many of the clumps of grass in the field were covered over by this drifting macroalgae, Polysiphonia, a species of Polysiphonia, a red drifting algae that lives in very low salinity waters, was very abundant in the grass bed at this site. Next slide, please. Now I'm going to show many slides that look somewhat similar to this, and these show nutrient concentrations over the course of the experiment. This first one I'm showing is for ammonium, and as you can see in the first 4 time points, which were before the nutrient addition, there were fairly low concentrations of this nutrient in all of the treatments. So it didn't seem to be that the grass presence or absence made much difference as far as the levels of ammonium early in the experiment. But then at the time that, next slide, please. Once nutrients were added, you get this incredible spike in nutrients in the nutrient-enriched treatments, whereas the unenriched treatments don't change that much. Next slide, please. Moving on to one of the other nutrients, this is nitrite. You can see that there's some differences in the pattern of this nutrient where it's actually declining for the first couple of dates. I assume as microbial life processes this nutrient into other forms or absorbs this nutrient. But then in the treatment where nutrients are added, you see an increase over time in this nutrient. Next slide, please. For nitrate, a similar pattern where there's an initial decrease in nitrate in the unenriched treatments, but then once the treatments are enriched with nitrate, the nitrate levels are just through the roof for the entirety of the remainder of the experiment. So it's a hyper-eutrophic environment was created in these tanks where we added nutrients. Next slide, please. For phosphate, you see in the unenriched treatments a steady decrease in the availability of orthophosphate over time, whereas the levels of total phosphorus don't change as much. So I think this just reflects sort of a transformation in what form the phosphorus is in by the plants and microbial life in the mesocosm tanks, whereas in the tanks where the fertilizer was added, the phosphorus levels went through the roof and stayed there for the remainder of the experiment. Next slide, please. In terms of phytoplankton, the algae in the tanks, there was some variability in initial conditions, perhaps due to just sort of the stochastic effect of what algal propagules were included initially in the buckets. There was some variation in algal abundance initially in the buckets, but that sort of equalized early in the experiment, and there was sort of the steady low concentration of chlorophyll in the water column of the tanks until the nutrient enrichment. After the nutrient enrichment, as you might imagine, there was far more phytoplankton in the nutrient-enriched tanks than in the unenriched tanks. But for this variable, there was an effect of Vallisneria. In the tanks with Vallisneria, and I need to click here, in the tanks with Vallisneria, which are the green bars, they were always lower than their corresponding white or gray bars, indicating that with the same level of nutrients, either ambient nutrients or enriched nutrients, there was always less phytoplankton in the presence of Vallisneria than there was without Vallisneria. So this was consistent with our initial hypothesis that an ecosystem service of Vallisneria would be to suppress phytoplankton growth. This occurred without a major reduction in nutrients in the water, though, so we think that the mechanism by which the grass was reducing the phytoplankton growth was not by competitive nutrient uptake in this case, but perhaps by some allelopathic effect, an algae suppressing chemical that was released by the Vallisneria. Next slide, please. This is a visual of what the tanks look like that shows the obvious difference between the panels on the left, which had no extra nutrients added to them, and the panels on the right, where we had this hyper eutrophication by nutrients. But you can also see a somewhat more subtle difference between the grassy tanks, which are on the bottom row, and the unvegetated tanks, which are on the top row. Even in the presence of nutrient enrichment, the grassy tank had less algae in it than the unvegetated tank. You can see how I hate to say it, but how pathetic the grass looks in these tanks. It started out small and it didn't grow very fast. We've had other plantings of this Caloosahatchee strain of tape grass in buckets and mesocosms at FGCU, and we found that it just takes a very long time to grow and increase in density. So if we run this experiment again, we may actually leave as long as a year for the grass community to establish in the tanks before we do the manipulation. That's something that we would do differently if we were rerunning this. Next slide, please. In terms of epiphyte growth, that's the attached algae that grows to the blades of seagrass, and we measured this on these artificial seagrass ribbons. We really didn't see much of this growing in our mesocosm tanks. This might be because we had stocked the tanks with snails for ecological realism, and the snails may have eaten some of that epiphytic algae. It could also just be that the phytoplankton that grew in the tanks dominated, and there wasn't much benthic algal growth. But in the field, there was quite a bit of algal growth. In fact, these ribbons were really fouled with stringy filamentous algae, and the mass of that was particularly high outside of the tape grass bed. So inside the tape grass bed, we did not see much epiphyte growth on the artificial seagrass ribbons, but outside the seagrass bed, we saw a lot. And the mechanisms of that we're still trying to figure out. Next slide, please. These results show the change in biomass from the initial to final dates of the mesocosm experiment and the change in shoot numbers from the initial to final dates of the field experiment. This isn't quite an apples to apples comparison because we're looking at biomass changes over the course of the experiment on the top panel and shoot number changes in a different experiment on the course of the bottom panel. But one of the points that I wanted to make from this was that probably due to the shock of being transplanted, there was this sort of initial decline in biomass of grass in the tanks. And in order for SAV to have the best chance of absorbing nutrients from the water, it would really need to be in an active growth period where dissolved chemicals in the water are turning into a large growth of grass. So it may be that we would have needed to run this experiment quite a bit longer in order to get enough grass growth to sequester a substantial amount of nutrients, versus in this case, there was actually this sort of decline, which may have been seasonal or transplant shock in the experiment. And although the grass survived, there was actually less biomass at the end of the experiment than the beginning. In the field, though, we were seeing an increase in grass abundance during the course of the experiment. So that's a good sign from the field experiment. Next slide, please. One of the most interesting results that we did get from the mesocosm experiment, despite the problems that we had and our regrets about the level of nutrients that we had added, was we saw a really big difference in the tissue nutrient concentration of the grass. Every living thing is made up of carbon, hydrogen, oxygen, nitrogen, phosphorus, and some other chemicals. But in plants, the ratio of those elements can vary depending on their availability in the environment. And the carbon to nitrogen ratio in particular is quite flexible in many plants. If they're growing in a nutrient-enriched environment, they can take up extra nitrogen, and this is one of the features of these plants that we think might allow them to sort of help fight eutrophication and absorb excess nutrients from the environment. And we saw that the Vallisneria in these nutrient-enriched treatments had about twice the nitrogen content as the Vallisneria grown with ambient nutrient concentrations. And this was true in both the blades of grass and in the roots and rhizomes of the grass. They were both richer in nitrogen where they had a surfeit of nitrogen in the water column to absorb. Next slide, please. Another difference that we saw between the grass that was growing in nutrient-enriched conditions, which is the bars on the right, and ambient conditions, which is the bars on the left, was that there was a lower shoot to, well, let's see, there were more roots relative to shoots in low nutrient conditions, whereas in high nutrient conditions, there was less root relative to shoot. This is a change in plant morphology that's pretty common as a response to nutrient enrichment, where if there's plenty of nutrients that they can absorb from the water column, they don't grow as large a biomass of roots. Next slide, please. So the final thing that I wanted to do with these data was try to extrapolate them to the capacity of Vallisneria to absorb and hold on to nutrients in the environment. For this, we had to combine some of the data from the experiment with some mapping and monitoring data that we had from the field. And this figure shows bathymetry layer that we used in association with there's a cheer camp or something happening in the quad of the university here. I apologize if that's loud. You can see this sort of rough bathymetry layer that we have for the Caloosahatchee, and the yellow area along the shoreline is supposedly the 0 to 3 feet depth. And this is the potential habitat for Vallisneria based on the light conditions in the Caloosahatchee. Our sort of best hopes for restoration recovery of Vallisneria is that this whole area will be filled in with Vallisneria. And so we use that to sort of get our area estimate for what we could restore. These little white squares mark points where we began transects we began and completed transects in 2020 in our survey of the seagrass in the estuary. And so we combined our seagrass survey data with our estimate of the area surveyed to figure out how much Vallisneria there is in this part of the estuary now. And then we combined that with some data that we had on the weight of a seagrass shoot and the nitrogen content in a seagrass shoot from the tissue nutrient analysis that we did for this project to figure out how much nitrogen was locked up currently in the shoots of Vallisneria in the estuaries. Next slide. So this slide shows the math that we did. And I'm sure we'll revisit this. We have sort of the general equation, but I'm sure that we'll tweak the values and check them against all sorts of references, get our estimate as refined as we possibly can. But this is our first shot at coming up with the statistic for how much nitrogen is held in the grass of the Caloosahatchee Estuary and how much could be retained by that grass if we restored it to a full density of 434 shoots per meter squared within all of this shallow shoreline area of the upper Caloosahatchee Estuary. I was a little disappointed, honestly, in how low these final numbers were. Only 3 to 7 kilograms of nitrogen are held currently in the very sparse patchy grass of the upper estuary. So this is really kind of a pittance compared to the nutrient loading in the estuary, and suggests that we're a long way from having substantial nutrient sequestration by the grass that's in the estuary. If we do this full restoration and go from 4.7 shoots per meter squared on average in this part of the estuary to 400 something shoots per meter squared on in this part of the estuary, then we could get hundreds of kilograms of nitrogen, which is better, but still kind of low. So let's go to the next slide. I have some final thoughts on this. So the amounts of nutrients that are removed from the water column and stored in the living plant tissue itself is kind of inherently a low-end estimate of the plant's contribution to nutrient sequestration and water quality improvement, because nutrient sequestration isn't merely by the absorption into the living tissues of the grass, but by storage of organic matter in the organic rich sediments that tend to accumulate in dense seagrass beds. Phytoplankton and detritus drifting through the estuary or should accumulate on the bottom in dense seagrass beds and build up. And that is an area where a lot of nutrients can be stored. When we are done analyzing the sediment nutrient data from the vegetated and unvegetated areas of the Caloosahatchee field experiment that we just did, we may be able to add this sediments aspect of the nutrient sequestration into the equation and beef up our number for the beneficial nitrogen sequestration value of the nutrients. Another thing that can happen in the sediments under a seagrass bed is removal of nitrogen from the system by denitrification. In wetland and submerged aquatic vegetation systems, this wonderful microbial process called denitrification happens, which turns the nutrient forms of nitrogen into harmless nitrogen gas. And if a lot of that is occurring in the sediments beneath the grass as compared to the bare sediments, then that's another way that the restored seagrass could be contributing to nitrogen removal in the system, and we hope to get at that in the future as well. The other thing that may be occurring in the field, as it appeared to occur in our Mesocosm experiment, is suppression of algal growth, not so much by competitive uptake of nutrients, but through this allelopathic chemical interaction. And that could be really important for preventing harmful algal blooms in the estuary. This upper Caloosahatchee Estuary, as we know, is really susceptible to toxic cyanobacterial blooms like Microcystis aeruginosa, which was happening concurrently with the big red tide of 2018. And perhaps if we have healthy communities of Vallisneria, it'll be able to suppress some of the growth of that cyanobacteria. So that's another thing that we hope to investigate more in the future. This, I believe, is my last slide, so I'll be happy to take questions and comments now.
I got a question. If I may.
Yes.
If I may. There's
Certainly.
feedback. So, but I'm trying to avoid that. The only question I have is about the you call it epiphyte. On top of the blades of the valve, you'll find a lot of growth of different algae, different marl, different type of and we call it periphyton or aufust. It's just layers and layers of different. Have you determined that? Because that's really important. We did notice that in another type of plant, Nias guadalupensis. We were doing studies here a long time ago, but I think that is a major sequestration mechanism that the plant has that you should put into that in this equation.
Absolutely. So we included epiphytes in our solid materials nutrient analysis. So we measured the mass of the epiphytes, and we measured the tissue nutrient composition of the epiphytes to see how much of the nutrient absorption by the seagrass bed was happening in the epiphytes. In the Mesocosm experiment, it didn't work very well because there was so little accumulation of epiphytes in the Mesocosm experiment, which is interesting because in the field, there's quite a heavy accumulation of epiphytes. So we're looking forward to getting the results back from Florida International University's blue carbon analysis facility on the nutrient concentrations in the epiphyte material from the field experiment, which will probably give us our most realistic estimate of how much those epiphytes are doing to sequester nutrients. And that's another thing that we expect will increase our estimated amount of nutrients being absorbed by the restored grass beds in the Caloosahatchee.
Hey James, this is Dave Seeley.
Hi, David.
Hey, I just sent a report to Sea and Shoreline and Angler Action Foundation. And it includes the monitoring that we did just up until July 28th. I sent you by email some photos. What we're seeing, and we have 500 exposure cages in the river now. And what we're seeing is that the blades inside the cage are growing to greater than 1 meter in size. So I have to disagree with the statement that the strain in the river only grows so high. It appears that grazing pressure is really limiting the growth. I just sent a couple of photos to you by email, and I copied Jennifer Hecker as well. Another thing we've noticed over the years, and I've done studies for 19 years on tape grass in the Caloosahatchee. One of the things we've also noticed is that, yes, when the salinity gets high, the plants only grow so tall, they get really stiff bladed, and probably to reduce osmotic pressure. But I just wanted to share that we have also had densities of greater than 1,000 shoots per meter inside cages in previous studies, and I could share those with you if you like.
Yeah, so that's great. That's very encouraging. If we can get 1,000 blades per square meter, that certainly increases our estimate of the total amount of nutrient sequestration. We use this figure of 400 per meter squared from previous monitoring efforts that had sort of associated that level with 100% cover. But yeah, the density of the grass can get higher than 400 per meter squared. And obviously, taller plants would sequester much more nutrients. I think one of the things in our back of the envelope calculations that are giving us such a disappointingly low amount of estimated nutrient sequestration is the average size of our plants, which are so small. And if we can sort of unlock that key to helping this strain reach its full potential, which may have something to do with overwhelming the grazers or getting water quality conditions just right, then the amount of sequestration per plant part of the equation would go up a lot, and the total nutrient sequestration would go up a lot as well. It is really interesting. Not only it is really interesting how this one plant under different conditions, a different amount of grazing pressure inside a cage, outside a cage, can undergo such a transformation from that Lilliputian form to the really impressive form in the picture that you just sent. And we've seen in our other bucket experiments, great. In our bucket experiments with this, that it takes time in a mesocosm setting for this to reach that size. So we're not done with these mesocosm experiments with these plants. We're as always, the first time you do a study, you think of 1,000 ways you could have done it differently or better, or follow-up studies. And so we've already begun this one in the buckets. It just seems that it takes a while for the roots to develop their networks beneath the sediment, and for the microbial symbioses and everything to get cranking, and these things to sort of really grow. But they do eventually, reach the top of the tank and become lush like this. It just takes some time. So if we can get some measurements of nutrient sequestration under those conditions, we may be able to share a little more optimistic estimate with the public about the capacity for these things to absorb nutrients.
Yeah, thank you. I think it's encouraging. What we're looking at here in the slides show the samples that were collected on July 28th. The top photo shows the rock star was the short bladed one. These are outside the cages at Buzzard's Roost Island. And on the right is the green wave, which is a local variety that historically came from the river. And you can see the difference in the biomass and the size of just a couple of plants. The bottom slide or the bottom photograph shows on the left plants inside a cage at Site 2, which is upstream of Highway 31. And then on the right are plants at Site 3. The short-bladed plants are very dense mat of roots, solid carpet of roots, and very short plants that are being grazed upon. So, we've seen freshwater fishes, tilapia, freshwater turtles, manatees, ducks, everything seems to like to eat tape grass. But, inside the cages, we're able to get these large plants and they're flowering, which is another thing that's a problem. The grazing is not allowing the plants to reach maturity so that they're not producing the female flowers at the top of the plant that typically, are pollinated at the surface. So that's one of the problems we need to get the seed bank back in the river to really get the plants to take off and to get to that tipping point. That's in my opinion anyway. Thanks, James.
Yeah, those are some great points, David, and it definitely seems that reaching this sort of threshold needed for a self-sustaining system is the goal that we're all going for. And I just wanted to relate that back a little bit to the concept of ecosystem services and some of the features of the environment that can enhance those ecosystem services. One of the things that other researchers have shown to enhance the ecosystem services provided by the environment is biodiversity. And while we're only dealing with one species of plant here, there's a lot of biodiversity genetically within this one species of plant. And it's likely that the series of salinity intrusions that killed off most of the plant in the Caloosahatchee Estuary have reduced its genetic diversity, and the remaining seagrass is sort of representing just a small fraction of the historic genetic diversity of the plant. And it's great to see that in these restoration efforts, you're using multiple strains, which, as we sort of increase the genetic diversity, there'll be some strains that can tolerate some conditions better than others, and the overall sort of productivity and function of the grass should improve and become more like it was historically. So that's great to see.
James, I've actually got a question on the potential allelopathic response of algae from the Vallisneria.
Yeah.
Do you have any sources or literature that you can point to that provides more information on that, or other species?
Yeah, so there's someone in the talk here, Dana Detmar, is a former graduate student of FGCU who worked with my colleague, Dr. Serge Thomas, on floating plant islands in wet detention ponds and nutrient sequestration and other mechanisms of algal suppression by those floating plants. And Dana's thesis work showed something kind of similar to what we saw in the mesocosms, which was that the amount of algal suppression achieved by these plants exceeded anything that could be accounted for by nutrient sequestration alone. And there's sort of a process of deduction that led to this, maybe there's something else going on there. Maybe it's this chemical, allelopathic chemical investigation. So I know she looked into the research more deeply than I have to pursue that and find out what the possibilities were. But as far as what chemicals might be leaching out that are doing this, how different plant species might vary in their allelopathic algal suppression abilities, I am not very well versed in that yet. So.
Hi all. Dana here. So I, unfortunately, in my thesis defense, I was unable to have the opportunity to really tease out what exactly the particular chemicals were in the that were having the effect, but just that, we did see that some aquatic plant species were able to suppress algae, and what we found was that cyanobacteria was actually um, um, more susceptible to allelopathic chemicals than green algae. So there was greater suppression of cyanobacteria. There's a variety of literature out there that discusses what species of plants can secrete these allelochemicals. In my study in particular, it was canna and juncus that I investigated, and both of them had allelopathic tendencies. But I do have from my thesis a bibliography that I'd be happy to share if anyone was interested.
Thank you, Dana. I really appreciate the response, and I can probably reach out to you directly on that a little later on.